going to go ahead and record this meeting. And by recording this meeting, this will later be posted on to our
YouTube channel.
Our College of Health and Human Sciences Student Success Center YouTube channel. And we're going to go ahead and post the link in the chat for you guys by the end of this session, and Jason has posted it now, so that after this workshop, you guys can rewatch it as many times as you'd like on the YouTube channel. So let's go ahead and get started. So welcome everybody to the Biology 66 Human Physiology Final Exam Review Workshop. My name is Stacy Demertzis.
Hello everyone, my name is Jason Pang.
And we are tutors from the College of Health and Human Sciences Student Success Center. Go ahead, next slide.
All right, a little workshop overview. So in this workshop, we're going to cover key concepts from chapters 17, 18, 19, and 20. So we've already covered previous chapters in our previous workshop, and the videos are posted on our YouTube channel. So you guys feel free to go ahead and click the link and watch the previous workshops. But for this workshop, we're going to cover chapters 17, 18, 19. So for 17, we're going to talk about glomerular filtration, reabsorption of salt in water, and a little bit about countercurrent multiplier system.
And for chapter 18, which covers the digestive system, we're going to go over some specific structures and functions and really go in depth of the digestion of the 3 macronutrients.
And then for chapter 19, we're going to talk about metabolism, right? The two functional states, the absorptive and the post-absorptive states. And then we're also going to talk about diabetes mellitus.
And then we'll lastly go over chapter 20, which talks about the reproductive system. We'll give a brief overview of the definition of the reproductive system, and then an example of reproductive endocrinology.
Okay, keep in mind that your final exam will be this Thursday, May 20th, right? From 12:15 to 2:30 p.m. All right. Yeah, keep in mind that is in 2 days. So Thursday, May 20th, all right? Okay, a little practice question to warm you guys up to the polling. So when will comprehensive final exam be on? I'm gonna launch the polling. You guys have about 30 seconds to answer the question. Okay, so all of you guys got that correct? The answer is C, Thursday, May 20th. All right. Okay, let's start off with renal physiology. So renal, whenever you come across the word renal, think about kidneys, right? And everything that has to do with kidneys. Okay, so the term renal pertains to the kidney. Now, on the right-hand side, we see some of the basic structures of the renal system, right? So you can see the 2 kidneys, right? The kidneys are where blood volume is regulated and organic waste products are eliminated through formation of urine, where urine is produced, right? And the kidney also balances things like water and electrolytes, okay? And then we can also see the renal pelvis, right? So when urine is produced, it goes to the renal pelvis and it goes to the ureters. Ureter transfer the urine from renal pelvis to the bladder. Now the bladder is where urine is stored, right? I'm sure all of you guys know that. And the urethra is where urine exits our body, where micturition and urination occurs. Okay. So a little practice question. For micturition, where does the renal pelvis empty its contents? Where do they empty its content? Let me give you guys about 30 to 40 seconds for this. Okay, so we're good on that. All right, so the correct answer is C, the ureter, right? Let me go back to the previous slide. So urine is dumped through the renal pelvis, and after the renal pelvis, it goes to the ureter, right? There's no such thing as urinary duct, or and then the urethra is where urine exits our body, right? So for micturition, when urine is produced, it goes to the renal pelvis and then goes to the ureters. Okay, so the correct answer is C for this question. Now, which structure in the kidney consists of a renal corpuscle and tubule? What is that functional unit called? Try not to use your notes for this. So then, right, because you guys can't use your notes during the exam. So, do this to your best ability, to the best of your abilities. You guys have about 10 more seconds. Okay. So the correct answer is D, right? The nephron. Remember, the nephron is the functional unit of the kidney, okay? We're gonna talk more about that. So the nephron is the functional unit of the kidneys. It is a site where urine is produced, okay? And the nephron consists of the glomerular capsule or the Bowsman capsule right here. And it also consists of the glomerulus or the capillary network, the capillaries. And then there are also the tubules, right? The loop of Henle, the proximal convoluted tubule, and the distal convoluted tubule, right? This is the entire structure, entire unit of the nephron. And this is where urine is produced, okay? Now, there are about 1 million of these nephrons in our kidneys, so urine formation can practically occur anywhere, I guess you can say, in our kidneys. Now, here's the route of urine. Here's how urine is filtered and how it goes in the kidney. So blood first enters our kidney through the renal artery. Okay, so blood first enters our kidney through the renal arteries. Then it goes through more arteries, right? The segmental, the interlobar. You don't need to know these, but just know that it enters renal artery, and then it goes into the afferent arteriole. Okay, so from the renal artery, they goes into the afferent arteriole, and then they go into the capillaries, the glomerulus. Right now, in the glomerulus, about 20% of blood that travels through the glomerulus gets filtered into the Bowsman capsule or the glomerular capsule, right? 20% of that blood that travels through the glomerulus gets into the capsule, okay? Now, from the glomerular capsule, it's gonna go through more tubules, right? The loop of Henle, the proximal, the distal convoluted tubule, the loop of Henle, so on and so forth. And it gonna go into the collecting duct, right? And then the renal pelvis, and then the ureter, and then they're gonna go into the bladder, right? Okay. And keep in mind that about 20% of blood gets filtered into the capsule. Now, the remaining 80% of that blood that does not get filtered leaves the glomerulus through the efferent arteriole. Okay, so afferent with an A, an apple, right? A, then it means that that's where they enter the glomerulus. Okay, an efferent arteriole exit. Think about exit, where they leave the glomerulus. Okay. And then after the efferent arteriole, they leave through the peritubular capillaries, and then they go into our renal vein, and then where they go back into our heart. All right. Any questions regarding the nephron? All right. Let's talk about glomerular filtration, okay? So remember how I said that 20% of blood in the glomerulus gets filtered into the glomerular capsule, right? So glomerular filtration is the term for that. Glomerular filtration is a term that refers to the movement of solutes and fluid from the glomerulus into the capsule. Glomerular filtration, right? So let's take a look at this little graphic right here. Okay, so imagine the left side is the capillaries. Imagine the left side is the glomerulus, and the right side is the Bowsman capsule or the glomerular capsule. Okay, now fluid is filtered from the blood through these pores called the fenestrae, and these fenestrae are located on the glomerular side, in the capillary side. All right, so these large pores, right? And after that, they go through these filtration slits of the capsule side. Okay, so blood is first filtered through these fenestrae, these pores, and then it goes through the filtration slits, and then where they enter the interior of the capsule. All right, and this is the glomerular filtration. This is where they go. And keep in mind that filtrate of the glomerulus are mostly solutes, right? Like sodium chloride, and only a small percentage are proteins, a very small percentage. Okay. Any questions regarding glomerular filtration? So the key concept you should know is that like glomerular filtration is just a term for fluid and solutes moving from the glomerulus into the glomerular capsule. Okay. All right, a little practice question. What does glomerular filtration refers to? You guys have a minute. Is that about 10 more seconds? Okay, so the correct answer is E, right? Remember how I emphasized this point many times, right? Glomerular filtration is the movement of fluid and solutes from the glomerulus and into the Bowsman capsule, right? Right, they move from the capillary side into the capsule. That's called glomerular filtration. Okay, so the correct answer is E. Now we're going to talk about reabsorption of salt and water. Reabsorption. So what does reabsorption in the renal system refers to? And try not to use your notes for this, right? Trying to do this to the best of your abilities. Have 30 more seconds. Okay. So the correct answer is A. You guys all got that correct. All right. That's right. Reabsorption in the renal system refers to the return of filtrate from the tubule into the peritubular capillaries, right? The filtrate from the glomerular filtration gets filtered or gets returned back into the general circulation, right? So in the glomerulus and the capsule, right, around 180 liters per day are 100 liters of filtrate per day are filtered each day, are produced each day, right? Right, because 20% of blood that travels through this glomerular cycle, gets filtered in, right? And throughout the day, there are 180 liters produced. But only 1 to 2 liters of those are excreted via urine. Only 1 to 2 liters of those leaves our body, right? And the 99% of those return back to general circulation to maintain blood volume and blood pressure, right? And keep in mind that most absorption, reabsorption of salt and water occur in the proximal convoluted tubule, and some are also reabsorbed in the descending limb of the nephron, but mostly occur in the proximal, right, closer to the glomerulus. Now, here's the process of salt and water reabsorption. Here's how salt and waters are reabsorbed. So first, sodium is actively being transported. There are active transporters that pump sodium out of the tubule, right? So sodium is actively being pumped out of the tubule and into the peritubular capillaries, okay? Now, when sodium is actively being transported, chloride follows that movement passively because chloride is attracted to sodium, right? Because of electrical attraction, right? So when sodium is actively being pumped, chloride follows that passively. It's a passive movement. Now, when sodium chloride exits the tubule, it means that there's going to be an increased concentration of solutes outside of the tubule. That is the peritubular capillaries, right? So imagine sodium chloride from the tubule leaving and going into the peritubular capillaries. It increases the osmolarity. It makes it more hypertonic outside of the tubule. So when this happens, water is going to naturally follow that. Water is going to passively diffuses out of the tubule because of osmosis, right? Remember how water moves from an area where there are less concentration of solutes to an area where there is a greater concentration of solutes, right? Now in this scenario, sodium chloride moves to the peritubular capillaries and the water passively follows that because of osmosis. All right, and this is just the fundamental concept of reabsorption of salt and water. All right, that makes sense? Any questions? Any thumbs up? Nice. Okay. Let's talk about the countercurrent multiplier system. So what does the countercurrent multiplier system do? What is its function in the renal system? What is it responsible for ensuring? Read the answers carefully, guys. You guys about 10 more seconds, 10 to 15 more seconds. Okay. So the correct answer is B. Right? Most of you got that because it's correct. So the countercurrent multiplier system is responsible for ensuring hypertonicity of interstitial fluid surrounding the loop of Henle. We're gonna take a look how. So remember, the countercurrent multiplier system is a system that creates a hypertonic interstitial fluid surrounding the tubule. Like in other words, it increases the concentration of solutes outside of the tubule. It increases the osmolarity. It makes it more hypertonic. All right. But how does this happen? Well, it first occurs in the thick ascending limb. Okay. Now, in the thick ascending limb, sodium is first being actively pumped out. So sodium is actively pumped out of the tubule. Right? And remember, when sodium is actively being pumped, chloride just naturally follows that because of electrical attraction, right? So chloride is attracted to sodium. So now sodium chloride exit the tubule, or they moves out of the tubule, right? Now, water is impermeable here. Water is not permeable, so water cannot move out. But the interstitial fluid is hypertonic, right? Now, in the descending limb, water is permeable. So water can diffuse out. So water would diffuse out because the interstitial fluid created by the thick ascending limb is hypertonic, right? Because it's hypertonic, water diffuses out via osmosis. Okay. Now, when water leaves the descending limb, it makes the descending limb more and more hypertonic here, right? See, at the bend of loop of Henle, it makes it more salty. Okay. Now you can see where this is going, right? If the process repeats itself, if the process just keeps repeating itself, the osmolarity, the saltiness is going to multiply. Hence the name, the multiplier system, right? So the thick ascending limb makes the interstitial fluid hypertonic. Then the water leaves the descending limb, diffuses out of the descending limb. Then it makes the bend of Henle. It makes the descending limb more and more hypertonic. And when the process repeats, the osmolarity, the saltiness is going to multiply. All right. Now, I know this is a very weird concept to grasp. It was a weird concept, a difficult concept for me to understand in the beginning, right? But the main thing you should know for the exam is that the countercurrent multiplier system is just responsible for ensuring a hypertonic interstitial fluid surrounding the tubule. All right. Does that make sense? Everyone? Any questions? Any thumbs up? Okay. All right.
All right, so now we're going to talk a little bit about the gastrointestinal system, otherwise known as the digestive system. Next slide. Okay, so the gastrointestinal system has several different functions. And in this workshop today, we're going to go over their functions in terms of what it does with the food that we eat. So whenever we eat food, there's a lot of things that go on. There's motility, digestion, secretions, absorptions, and storage and elimination. All of these things that happen within our digestive system organs that helps us process the food we're eating. So let's go over these main functions. So motility. Motility means movement. So a motility function refers to the GI tract, which the GI tract is shown here in this image of the digestive system. It's GI tract peristalsis. And so peristalsis is a special vocabulary word that pertains to a lot of the motility function. Can someone define for me? Feel free to unmute yourself or put it in the chat. What does peristalsis mean? What's the definition of peristalsis?
Are they like wave-like contractions?
Yes, absolutely. So peristalsis refers to, as you said, the wave-like contractions of the muscles within the GI tract. So our GI tract organs, there's musculature within the organs. And so when these muscles produce this wave-like contraction, it propels our food down our GI tract so that when we eat our food, it can continue to move forward. Absolutely. So as food moves throughout our GI tract, the food needs to be digested. And so there's different kind of steps and different ways for food to be digested. So there's mechanical breakdown, which refers to the physical breakdown of food, like chewing, breaking up the food into smaller parts. And then the chemical breakdown is like changing the chemical composition of the food and really breaking it down so that we can later absorb those nutrients that are within the food. Secretion could be a little confusing. It was confusing for me when I was taking the course, but it refers to any type of exocrine or endocrine substances that are released from various organs. They're released into the GI tract to aid in digestion. And we're gonna dive into that further when we go over the specific organs. Once food is digested, as I mentioned earlier, we want to absorb those nutrients into our bloodstream. And so can somebody tell me, why would these nutrients want to be absorbed into the bloodstream? What does the bloodstream do with nutrients? Feel free to unmute or put in the chat. So I'm seeing here, transport it to cells around the body. Absolutely. So the cardiovascular system is a transport system. So when nutrients enter the bloodstream, the blood is going to then deliver these nutrients to all these other parts of our body so that we can use them, because we need nutrients to be healthy and survive. So once nutrients are absorbed, after we've digested the food and whatnot, we are going to then create waste. And we're going to learn a little bit more about like which organs are responsible for that. And so waste is kind of another way of explaining things that are indigestible. So we're going to create the waste, we're going to store it, and eventually we're going to eliminate it. Next slide. So I have a question for all of you. So this is more about anatomical identification of the digestive system. So, and it's more of a vocabulary question too. So I want to ask you all, what is the difference between the alimentary canal and the accessory organs? And this is a select all that apply question. So think about the definition of the alimentary canal, think about the definition of accessory organs, the identification, what are they? And then think about their role in the digestive system. Give you guys about 30 seconds. Select all the correct statements. About 10 more seconds, and then we'll go ahead and close the poll and review the answer. Okay, we'll go ahead and end the poll. Okay, so the answer is A and B. So the alimentary canal is also known as the pathway of food. So it's also known as like the GI tract. So it's all the organs that start from the mouth all the way to the anus, in which our food comes in contact with. The accessory organs are, we'll define those in a minute, but basically they don't come in contact with food. They're not a part of this tubular tract in which the food travels. So next slide. So similar question, what is the difference between the alimentary canal and the accessory organs in terms of like, what do they consist of? What are the organs that make up the alimentary canal? And what are the organs that are identified or classified as the accessory organs? And this is also a select all that apply. Give you guys about 30 seconds. About 10 more seconds. And then we'll go over the answer after we close the poll. Okay. So the answer is A and C. So the alimentary canal consists of the mouth, esophagus, stomach, small intestine, large intestine, rectum, and anus. And then the accessory organs consist of the liver, gallbladder, and pancreas. So when we think back to our last polling question, and we were identifying like the difference between like the pathway of food and organs that don't come in contact with food, now that you guys have been able to correctly identify these organs, we notice that yes, food comes in contact with our mouth, it travels down our esophagus, it goes into our stomach. It doesn't physically come in contact with things like the liver, the gallbladder, the pancreas. That doesn't mean they don't have an important role in digestion. It just simply means that food is not passing through them. Next slide. So just a brief overview and a visual to kind of show both the alimentary canal and the accessory organs. when we talk about the pathway of food through the digestive system, it really comes down to being able to anatomically identify them correctly. And then when we go into more like gastrointestinal physiology, that's when we really dive into understanding how is food digested? What is going on within these organs that can accomplish the goal of the digestive system? So some questions I want you guys to think about is when we review the details of the digestive system is, where does digestion start? And later on, where does optimal digestion occur? And where does digestion end? Think about those like key points in the digestive system, like what organs are responsible for such. So, and we'll go over those. Just think about that in the back of your mind when you're reviewing GI physiology. Next slide. So just like a quick overview of the pathway of food through the GI system, and also keeping mind of the digestive functions. So here I have the alimentary canal organs. So we start at the mouth, food then travels down the esophagus, it then reaches the stomach, it will then travel into the small intestine, it will then travel into the large intestine, reach the rectum, then the anus. And so with respect to these organs, there's a certain responsibility that these organs have. So the mouth is actually where digestion of food first occurs. And then the esophagus is going to then take that chewed up food and is going to travel or transport it down into the stomach, where digestion of food is going to continue to occur. And then once it reaches the small intestine, digestion of food is going to continue as well. And that's actually where the optimal digestion occurs. That's where digestion finishes off, is in the small intestine. And that's where then the absorption of nutrients is going to begin. And then content is going to travel into the large intestine, also known as the colon. And the large intestine is where absorption of water and the creation of waste is going to occur. And the rectum and the anus are responsible for then taking that waste that was created and transporting it out of our body. So a question I have for you, we're seeing the terms a lot like digestion, transportation. A common concept within GI physiology is reminding us, what is the function of the esophagus? Is it digestion or transportation? Feel free to put it in the chat. And Jason, I'm going to have you annotate because I am actually unable to annotate. I don't have the permissions to. So if you can type in the answer. Thank you. Yes, and I'm seeing in the chat transportation. You guys are absolutely correct. So a lot of students get confused because they're like, well, if digestion starts in the mouth and it's also happening in the stomach, does that mean it's also happening in the esophagus? No, the esophagus is just a transportation tube. So absolutely, you guys got that correct. And thank you, Jason, for adding in that answer. So go to the next slide. So let's talk about digestion in terms of the nutrients that are going to later be absorbed, the nutrients that are found within our foods that we're going to break down so that our body can use it to be healthy. So my question for all of you, and we'll go ahead and launch the polling question, are what are the 3 macronutrients? Give you guys about 30 seconds.
And Stacy, I think you'll be able to annotate now. I think I just changed the settings a little bit.
Yes, I see that now. Thank you. Okay. Guys, about 10 more seconds, and then we'll go over the answer. Okay, yes, so the answer is D. So carbohydrates, proteins, and lipids are our 3 macronutrients. The reason why A, B, and C are incorrect is because they contain answers like the minerals and the vitamins. Minerals and vitamins are known as the micronutrients. And basically the difference between the two is that we need large amounts, macro means large, we need large amounts of carbs, proteins, and fats we don't need large amounts of vitamins and minerals. So that's the main difference. So next slide. So let's dive into how these macronutrients are digested within our digestive system. And in this slide, we're going to focus on the alimentary canal, the organs such as the mouth, the esophagus, the stomach, small intestine, large intestine, rectum, anus, right? The pathway of food, the GI tract. So we're going to focus on which nutrient is digested where. So let's start with the mouth. So in the mouth, we have things called salivary glands, and salivary glands secrete salivary amylase. And salivary amylase is an enzyme. And a way that we can remember that this is an enzyme is it ends in ase. So ase means enzyme. So if you see that word, that's a vocabulary clue. So salivary amylase is going to help digest which macronutrient in the mouth. Can somebody tell me, either unmute or put it in the chat? Okay, I'm seeing in the chat carbohydrates. Yes, absolutely. So carbohydrates they start to be digested in the mouth. So as those are starting to be digested in the mouth's responsibility is to mix food and saliva, and it's going to create a bolus. And basically, bolus is a fancy name for chewed up food. So that's what's occurring in the mouth. Let's move on to the stomach, because remember, the esophagus is then going to transport that bolus into the stomach. So within the stomach, digestion is going to continue to occur, and something called chyme is going to be formed. And chyme is basically a mixture of this bolus and gastric juice. The word gastric means stomach, and juice is what's secreted. So chyme is formed. So what else is going on in the stomach that's going to promote digestion? Well, hydrochloric acid is going to be secreted by the parietal cells. And what hydrochloric acid does, it's going to create an acidic environment to digest which macronutrient. What macronutrient is optimally digested in an acidic environment? Let you guys answer in the chat. Feel free to unmute yourself too. So I'm seeing a lot of answers in the chat saying proteins, and that is absolutely correct. So proteins like to be digested in an acidic environment. Hydrochloric acid is acidic because it's an acid and it's secreted into the stomach. So proteins are going to begin to be digested in the stomach. Also, chief cells, another type of cell, are going to secrete pepsinogen, and pepsinogen is a precursor to pepsin. And pepsin is an enzyme that digests what? What macronutrient is going to be digested by pepsin? Yes, and I'm seeing in the chat that is proteins as well, and you guys are absolutely correct. So proteins are going to be digested, or at least start to be digested, within the stomach. So now that food has traveled the mouth, the esophagus, the stomach, now we're going to enter the small intestine. And the small intestine is where it's going to finish the digestion of all macronutrients. And since we haven't gotten to lipids yet, that's actually where lipids, otherwise known as fats, are going to be digested. It's going to start being digested and finish its digestive journey within the small intestine. And these things called brush border enzymes, which are located in the microvilli. If you were to examine a small intestine under a microscope, you'll notice these like finger-like projections. They're called microvilli. They're within the mucosa of the small intestine, and they have these little enzymes on top of them that help digest the macronutrients. And so once all the macronutrients are digested, absorption is now ready to occur, and absorption occurs in the small intestine. So we'll go to the next slide. So that concludes talking about like the alimentary canal, like what goes on within the organs of the alimentary canal to promote digestion. So now let's take a look at how our accessory organs, our liver, our gallbladder, our pancreas, what role do they play in digesting our food? So when food comes in contact with the mouth, it's primarily like the salivary amylase. There's no actions from the liver, the gallbladder, the pancreas. Nothing, no actions yet are occurring from these accessory organs. Same within the stomach. The action starts happening when food reaches this small intestine. So let's talk about what's going on. So bile. Bile is a substance that is secreted by one of the accessory organs. Can somebody tell me which organ that is? Where's bile created? Yes, and I'm seeing in the chat the liver, and that is absolutely correct. So that is the accessory organ that creates bile. And so once bile is created, it's secreted, where is it going to be stored? Which accessory organ stores bile? I'm seeing in the chat gallbladder, and that is absolutely correct. And if we look at this image here, we see the liver here in red and then the gallbladder here in green. And we can see that they're very close to each other. So the liver will then, as you guys answered correctly, create the bile and then store it in the gallbladder. It's going to be stored there until food's going to come down and it's then eventually going to be used. So as bile is stored in the gallbladder, it's going to be released into the small intestine. That's what SI, the abbreviation, refers to, small intestine. It's going to be released by a certain duct. What duct is that called? What duct transports bile from the gallbladder to the small intestine? Feel free to put it in the chat or unmute yourself. Yes, so I see in the chat the answer common bile duct, and that's absolutely correct. So the common bile duct will then transport that bile, and it's going to help digest which macronutrient. And here's a hint. Think about the vocabulary word emulsification. And so emulsification basically means it's going to break down a substance before digestion. It's kind of like helping the substance be broken down into smaller components to make digestion easier. So which micronutrient is going to be digested here? What does bile help digest? And I'm seeing in the chat, you guys are saying lipids, and that's absolutely correct. So bile is a fat emulsifier. It helps break down that fat further so that it can be better digested. So that's what the liver and the gallbladder do. Now let's talk about the pancreas. So the pancreas has a few enzymes: pancreatic amylase, pancreatic trypsin, and pancreatic lipase. And similar to the common bile duct, these enzymes that are formed in the pancreas are going to be secreted into the small intestine by another duct. What duct is that called? Which duct travels pancreatic enzymes into the small intestine so that they can help digestion? Feel free to unmute or put it in the chat. Yes, so I'm seeing in the chat the pancreatic duct, and that's absolutely correct. So the pancreatic duct will then transport these enzymes. And so for these three enzymes, which enzyme, so pancreatic amylase, trypsin, and lipase, which enzyme digests which macronutrient? So there's three enzymes listed and there's three macronutrients. I want you guys to match those. So what does pancreatic amylase digest? Which macronutrient? So I'm seeing carbohydrates, and just to save some space, I'm going to put carbs. You guys are correct. And then what does trypsin help digest? It's formed in the pancreas, delivered to the small intestine, and it's going to it's going to digest, yes, absolutely, proteins. And then pancreatic lipase, what is that going to digest? Yes, absolutely, lipid. Oh. Lipids is the last answer. So absolutely, you guys got all that correct. So we'll go on. So that's how the accessory organs help aid in digestion. Go to the next slide.
Okay. Sec. Okay, so now we're going to talk about regulation metabolism. All right. So there are two functional states for providing energy for our cells, the absorptive and the post-absorptive state. Now, we're going to talk about the two functional states, including diabetes mellitus. So first, let's talk about the absorptive state. Which of the following processes occur during the absorptive state? Think about anabolism. What is anabolism? Is it synthesis? Are we breaking down food or are we synthesizing food? And remember to read the questions carefully, right? The absorptive state. What occurs during the absorptive state? Not the post-absorptive, but the absorptive. During the feasting stage, right? You guys have about 30 more seconds. Let's have about 15 more seconds. Okay, so the correct answer is D, right? Lipogenesis and glycogenesis occur during the absorptive state. Gluconeogenesis occur during the post-absorptive state. So the correct answer here is D, all right? We're gonna take a closer look at this. So the absorptive state is the 4-hour period following a meal, right? It's a period after a meal and it's about 4 hours long. Now, this is a state of anabolism. Right? Can anybody tell me what anabolism means? Like, what is anabolism? You can just type in the chat or unmute yourselves. What is anabolism? Is it a synthesis of something or are we breaking down something? Okay, so anabolism is the synthesis of energy reserves, right? So when we eat food, when we consume food, our body make energy reserves out of the nutrients that we eat, right? So absorptive state is characterized by anabolism, when we make energy reserves. So when we first eat carbohydrates, it gets digested and absorbed into our bloodstream as monosaccharides. Okay, mono means single, one, right? It's just one chain. So it's the most simple form of glucose, right? Glucose and sugar. So I guess you can say that carbohydrates gets absorbed in the bloodstream as sugar, right? Now, with the increase in glucose concentration, insulin is secreted to promote the cellular uptake of glucose, right? So when insulin is secreted, glucose enters our cells so that it can be used or it can be stored for later use, right? And if our cells doesn't need to use glucose for energy, then it'll be stored as glycogen or triglycerides or fats for later use, okay? Now, in our skeletal muscles, glycogen are synthesized from glucose. So glycogenesis is the process in which glycogen is synthesized from glucose. All right, so glucose is converted to glycogen, which is a storage form of glucose. It's stored, right? And it occurs in our skeletal muscles. Right? That's one way how glucose can be stored. Now, lipogenesis is the process in which glucose is transformed to triglycerides or fat. Okay. Now, in our adipocytes, it occurs in our adipocytes, in our adipose tissue cells, right? So triglycerides are the storage form of fat, right? But glucose can also be converted to triglycerides because from the molecule acetylcoenzyme, okay? And it occurs in our adipose tissue cells, right? So these are the two processes in which glucose can be synthesized for energy reserves, okay? Now, obviously, when glucose enters our cells, when it is stored, that means there's going to be a decrease in plasma glucose, right? Because when we eat carbohydrates, glucose concentration is increased. Now, insulin is secreted when there's an increase in glucose concentration. So when cell uses our glucose or when they synthesize new energy reserves out of glucose, then there's a decrease in plasma glucose, okay? Now, when we eat lipids, like things like fats and vitamins, they're absorbed into our bloodstreams as chylomicrons. Okay, lipids are absorbed as chylomicrons and is stored as triglycerides. Okay, so triglycerides is a storage form of fat, but glucose can also be stored as triglycerides. Okay, fatty acids can be used for energy. Okay. So the key concept in during the absorptive state, during the feasting stage, is that anabolism occur, right? So absorptive state is characterized by glycogenesis and lipogenesis, where energy reserves like glycogen and fats or lipids are synthesized and stored. Okay, this is absorptive state. Okay, so a little practice question. During the absorptive state, which hormone promotes the cellular uptake of glucose? Is it glucagon? Is it insulin? Is it glycogen? What is that hormone called? And this is during the absorptive state. I'm gonna give you guys about 30 more seconds. Okay. So the correct answer is C. Insulin, right? Insulin is secreted when there is an increase in plasma concentration of glucose, right? And we don't want too much sugar in our blood. We don't want too much glucose in the blood. So insulin is secreted so that glucose can enter our cell, right? It promotes a cellular uptake of glucose. So there can be less glucose in our blood. We can't have too much sugar in our blood or else we have diabetes, right? So the correct answer is C. Now we're going to talk about the post-absorptive state. So during the post-absorptive state, what happens to blood glucose that stimulates the secretion of a certain hormone from the pancreas? And the key word here is post-absorptive, all right? Not absorptive, but post-absorptive during the fasting stage. Give you guys about 15 more seconds. Okay. So most of you guys said C and some of you guys said B, right? While B is correct if the question was during the absorptive state, but in this question, we're talking about the post-absorptive state, right? When there's a decrease in blood glucose, when we're not eating, when we're fasting, that stimulates the secretion of glucagon from the pancreas, right? Yes, increased blood glucose do stimulate secretion of insulin, but we're not talking about the absorptive. We're talking about post-absorptive, okay? Now, so the post-absorptive state is after the 4-hour long of absorptive state, when the GI tract is essentially out, when the GI tract is essentially empty of nutrients and no longer synthesize energy reserves. Okay, and it's also known as the fasting stage. Okay, now during this period, catabolism occurs. Can somebody tell me what catabolism is? Like, are we still synthesizing or are we still breaking down? Is it synthesis or is it breakdown? Right. So catabolism is the breakdown of our energy reserves, is the utilization of our energy reserves. Right. So when our body uses our own energy storage to supply and produce energy. Okay. So obviously, when our body is fasting, right, when we're not eating any food, there is a decrease in plasma concentration of glucose, right? Now, there are many different ways in which our body can maintain and increase blood glucose, right? First, the secretion of glucagon stimulates the breakdown of glycogen. Okay, so glucagon is secreted when there's a decrease in plasma glucose. Right? Glucagon is opposite to insulin, right? Remember, insulin is secreted when there is an increase in plasma glucose, but glucagon is secreted when there's a decrease in plasma glucose. Okay, and now when glucagon is secreted, this process called glycogenolysis occurs. So glycogenolysis is the breakdown of glycogen, right? So whenever you see the term that ends in olysis, just know that it's the breakdown of something, all right? And whenever you see the term that ends in genesis, right, like glycogenesis or lipogenesis, it means the synthesis of something. That's anabolism. Now in catabolism, glycogenolysis means we're breaking down glycogen and we're using glucose to supply energy, for energy. Okay, so glycogen is broken down to release and produce glucose to increase plasma glucose. Plasma concentration of glucose can also be maintained and increased through two other processes called lipolysis and gluconeogenesis. Right. So many of our tissue cells can use fatty acids for energy. That is why glucose can be stored and fats can be stored as triglycerides. So we can use it for energy later on. Right. Now, in the process of lipolysis, triglycerides is broken down. Okay, so lipolysis is the breakdown of lipids, or the breakdown of triglycerides, to release fatty acids and glycerol so our body can have energy, right? Okay. Now, in gluconeogenesis is when our liver and kidneys make new glucose. Okay. So gluconeogenesis is another process in which our body can increase blood glucose, right? Think it's genesis. Genesis means synthesis of new glucose. Right? And we're synthesizing new glucose because there's a decrease in plasma glucose. We want to increase blood sugar, right? Okay. So keep in mind that during our fasting stage, insulin secretion is decreased, but glucagon secretion is increased due to the decrease in plasma glucose, right? So during this stage, there's a release of fatty acids and glucose and amino acids into our blood so that our body can produce energy using those. Okay. Any questions about the post-absorptive states? Any thumbs up? All right, awesome. Okay. So which of the following processes is considered catabolism? Just launch the polling. Remember catabolism, you guys said it right, is the breakdown of something. It's a breakdown of, it's a breakdown process. Read the answers carefully, guys. You guys have about 30 more seconds. About 10 more seconds. Okay. So the correct answer is A, right? Remember how I said that olysis, when a term ends in olysis, it means to break down of something, right? So in catabolism, literally means the breakdown and utilization of our energy reserves. And all of these terms would not apply because the endogenesis, that means they're synthesizing new lipids, they're synthesizing new glycogen and ketone bodies. Now, so the correct answer is A, lipolysis, the breakdown of lipids. That's catabolism. Okay. And certainly a question like this will definitely be on the exam. Okay, let's talk about diabetes mellitus. So I'm sure you guys know what diabetes are, right? Is when there's a high concentration of glucose in our blood, or a high concentration of sugar in our blood. Diabetes mellitus is a chronic condition that affects an individual in the long term. It doesn't just occur in a week or in a few days or a month. It's a long-term condition. Another term to characterize this, to describe this, is hyperglycemia. Right? Hyper literally means excessive or a lot, and glycemia just means blood sugar, right? Blood sugar. So hyperglycemia literally means high blood glucose. So there are two types for diabetes mellitus. Type 1 diabetes is characterized by insulin deficiency. Okay, meaning there is a lack of insulin produced by our body, by our pancreas. Okay, there's a lack of insulin produced. Insulin deficiency is due to the autoimmune destruction of our beta cells of pancreas. All right, so our beta cells are destroyed by our autoimmune destruction of our beta cells so that they cannot make insulin. Right, that's type 1 diabetes. And this kind of autoimmune destruction is in a type 1 diabetic is caused by genetic and environmental factors. So it's a genetic issue, an environmental issue, right? That's type 1 diabetes. Now, on the other hand, type 2 diabetes is characterized by insulin resistance. Insulin resistance, meaning our target cells are not responsive to insulin in the way that they should, right? They're not responsive, they're resistant to insulin. That's in a type 2 diabetic. Okay. Type 2 diabetes is more likely to occur in overweight and obese individuals because insulin sensitivity is most likely to be reduced by excessive fat and adipose tissue cells, right? So in an obese individual, in an overweight individual, they have more fats and more adipose tissue cells, so their cells are less sensitive to insulin, right? Type 2 diabetes is the most common form of diabetes, right? 90 to 95% of individuals diagnosed with diabetes have type 2. All right. Are there any questions about diabetes mellitus? Give me a thumbs up or unmute yourself to ask a question. Okay. Little practice question. So is type 1 diabetes characterized by insulin resistance? And is type 2 diabetes characterized by insulin deficiency? Right? Is true or false? Oops. Ooh. All right, I'm gonna give you guys about 15 more seconds. Okay. All right. So the correct answer is B, right? Type 1 diet. So it's false. This statement is false, right? Because type 1 diabetes is characterized by insulin deficiency. All right, I'm gonna go back to the previous slide. Type 1 diabetes is characterized by insulin deficiency when there's a lack of insulin, and it's caused by genetic and environmental factors. Right? Type 1 diabetes is when our beta cells of our pancreas are destroyed, so then they cannot make any more insulin. Now, on the other hand, type 2 diabetes is when the insulin or target cells are resistant and unresponsive to insulin, right? Insulin is pretty much useless, right? Because our target cells won't respond to it. So for this statement is false because, right, the statement is pretty much flipped, right? Type 1 diabetes is characterized by insulin deficiency, while type 2 diabetes is characterized by insulin resistance, okay? All right. Talk about reproductive.
Yes, so we're gonna kind of briefly go over the reproductive system and provide an example. We'll talk a little bit about reproductive endocrinology. So the reproductive system. So by definition, the reproductive system includes sexual reproduction, which is where the genes of two individuals are randomly combined to form a new generation. So with reproductive endocrinology, there's this relationship between what's going on in our reproductive system and what's going on with our endocrine system to help our reproductive system function. So we wanted to provide an example from reproductive endocrinology. And so the example we provided refers to what happens during puberty when there is this increase in the release of our gonadal steroids, otherwise known as our sex hormones. So during puberty, the gonads, which are the ovaries in females and the testes in males, they increase their secretion of sex hormones. And how do they do that? What prompts them to do that? Well, it's a stimulation from going all the way back to the hypothalamus. So the hypothalamus is a structure within our brain. And so during puberty, the hypothalamus is going to release something called GnRH. So can somebody tell me what that stands for? What is that abbreviation referred to? Feel free to unmute, put in the chat. Yes, so it's the gonadotropin releasing hormone. So what happens is the hypothalamus is going to release this gonadotropin releasing hormone, and it's going to then trigger the anterior pituitary gland, which is another structure within the brain. And so as the hypothalamus releases this GnRH, it's going to trigger the anterior pituitary gland to then release two hormones, follicle stimulating hormone, otherwise known as FSH, and luteinizing hormone, otherwise known as LH. And these two hormones are known as the gonadotropin hormones. So it kind of makes sense that the gonadotropin releasing hormone is going to later trigger the release of the gonadotropins. So after the anterior pituitary gland releases the gonadotropins, it's going to then trigger the gonads. And as we explained earlier, the gonads are the ovaries and the testes. It's going to trigger those structures to then release sex hormones, testosterone, progesterone, estrogen. And these hormones are known as gonadal steroids. Also, it's going to prompt gametogenesis. And so gametes are sperm and eggs. So sperm is found within the testes, eggs are found within the ovaries. And so the synthesis or the genesis, of these cells are going to form as well. So this kind of sequence, these steps are what's known as like the 3 hormone sequence. And so I want you guys to tell me, where else have you seen this concept before, like a 3 hormone sequence? And what topic or what chapter does this remind you of? Three hormone sequence. What other examples have you guys seen of this? Doesn't have to necessarily relate to the reproductive system. Yeah, thyroid hormone production. What's coming to my mind is like aldosterone, antidiuretic hormone. So you guys learned about it. We wanted to provide this example of reproductive endocrinology to kind of synthesize some information and show that there's a relationship between what happens with our hormones and how that helps our reproductive system function. And we'll go. Any questions on this before we move on? Questions? Nope. All right. Well, thank you all. We have about 13 minutes. If any of you guys have any questions, we'll be happy to go over any questions you guys may have.
You guys can also share study tips with each another, right?
Yeah, we can talk about that too. They don't have to be like specific questions material. We can talk about study strategies, whatever you guys would like to ask or talk about. We have a bit of time to do that, so we can discuss. But thank you all for coming and participating.
I mean, one of the main strategies I would give you guys, one of the main tips, studying tips I would give you guys is to delete TikTok if you want to actually study, you know? I mean, TikTok social media is a big distraction, you know?
Yeah.
I know this is gonna be a cumulative exam too, so it's gonna be a little difficult. Never downloaded TikTok?
Nor have I.
You guys don't go on TikTok? How do you guys spend your free time?
Other than not going on TikTok, I'm gonna recommend, I'm a big fan of like repetitive studying. So like after you review a chapter and then you go into the next and then you go into the next, like don't stop there, like review it again and again and again. Even though it takes a long time, the more times you see the information, the better or the easier it is going to be to help memorize it. So even if you feel like you have it down, keep quizzing yourself, keep reviewing it. I always say go into exams like feeling over prepared.
And so to answer Vanessa's question, the 2 tips I would give you is for concepts like the diabetes mellitus, right? You can create a T-chart, a graph for you, a chart for you, right? So you can easily identify type 1 and type 2 diabetes. And also for like absorptive and post-absorptive states, where there's 2 different periods of metabolism, you can create like a chart for you to understand it better. And also when you write down notes, when you rewrite down notes from lecture, try to write it in your own words rather than just copy it word for word, like verbatim, right? You know, you want to like make sure you understand the concept and write it in your own words, so then you can, I don't know, this is, yeah, group reviews are helpful too, right?
I've been
Yes.
going over the labs too. That's been helping because it's more like it explains kind of what happens instead of just like each step, like in words, like you see it. So then, yeah, that's been helpful for me.
Yeah, definitely